Hi! Thanks for visiting my project. My name is Wendy Yang. I'm a fifth-year graduate student at the University of Washington, and I'm working on my PhD in the laboratory of Dr. Vincenzo Cerulli. My project focuses on generating new insulin-producing cells from stem cells. I personally study basic mechanisms of cell-to-cell communication in stem cells as they grow toward immature pancreatic cells and eventually insulin-producing cells. Type 1 diabetes, also referred to as juvenile onset diabetes, is a devastating disease that affects more than 1.25 million people in the US alone. There are about 20,000 new cases in children diagnosed every year. This results in national healthcare costs of almost $15 billion every year. Type 1 diabetic patients can no longer produce their own insulin. For example, when a healthy person eats a donut, insulin will be released from the pancreas, which acts as a key allowing that sugar to enter cells and be changed into energy. Type 1 diabetic patients don't have any insulin and thus cannot change sugar into energy. This eventually leads to many different complications. Currently, the only treatment option is multiple daily injections of insulin to keep sugar levels under control. My research focuses on the development of new cell replacement strategies that can restore insulin production in patients with type 1 diabetes. Specifically, my goal is to turn human stem cells into insulin-producing cells that can one day be used for transplant. Although many scientists are capable of producing these cells, there is a bottleneck in the efficiency of production. My experiments aim to increase the quantity of insulin-producing cells made from stem cells. To do this, my experiments aim at understanding and manipulating the function of a class of my favorite proteins called connexins. These proteins function by forming small channels across cells that stick to one another. Think of cells as adjacent rooms. Connexins act like a door between them. These channels are used by stem cells to exchange biochemical information and make collective decisions to either grow and expand or acquire specific characteristics, such as becoming a cell of the brain, the heart, the skin, or the pancreas. I hypothesize that manipulation of specific connexins during stem cell differentiation can help convince stem cells to become pancreatic cell types. In line with this notion, I recently discovered that stem cells treated with chemicals that increase connexin function exhibit a much higher propensity to become insulin producers when compared to untreated stem cells. In this picture, we see many more pancreatic cells, marked in green, in the treated cells versus the untreated cells. My discovery is important because it significantly increases the number of insulin-producing cells that I'm able to grow. This could lead to innovative new strategies for deriving the billions of cells we would need for future use in transplants. I am so excited about my recent discoveries, and I'm seeking your help in funding my studies. I'm confident that they will lead to innovative new strategies in the treatment of type 1 diabetes. Thanks for watching.